Imagine our DNA as a vast, intricate library. Each book in this library is a gene holding instructions for building and operating our bodies. Sometimes these instructions contain errors, like typos in a crucial manual. These errors can lead to diseases. This dream began to materialize with the discovery of a remarkable system called CRISPR-Cas9. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. It acts like a pair of highly precise molecular scissors that can find a specific spot in the DNA and make a cut. The CRISPR system was discovered in bacteria. Bacteria, like us, face threats from invaders, particularly viruses. Over millions of years, bacteria evolved a clever defense mechanism. When a virus attacks, the bacterium can capture a small piece of the virus's DNA. It then stores this viral DNA snippet within its own genetic code in these CRISPR regions. The Cas9 part of CRISPR-Cas9 is an enzyme, a type of protein. Think of Cas9 as the actual blades of the molecular scissors. The CRISPR part, with its stored viral DNA, acts as a guide. This guide, RNA, leads the Cas9 enzyme to the exact matching sequence in the invading virus's DNA. Once the target is found, Cas9 makes a precise cut, disabling the virus. Scientists realized that this natural bacterial defense system could be reprogrammed. This ability to target specific DNA sequences with such accuracy was revolutionary. The journey to understanding and harnessing CRISPR was a gradual one, built on decades of research into the microbial world. Scientists first noticed unusual repeating sequences in bacterial DNA in the late 1980s. These sequences were the clustered, regularly interspaced, short palindromic repeats. The interspaced parts were initially a mystery. It was later discovered that these spacer sequences often matched DNA from viruses. This was a crucial clue. The idea of an adaptive immune system in bacteria began to take shape. Further research revealed that these CRISPR loci worked in conjunction with a set of genes called CRISPR-associated genes, or CAs genes. These genes produce the CAs proteins like Cas9, which are the workhorses of the system. When a new virus infects a bacterium, specialized CAs proteins snip out a piece of the viral DNA. This piece, called a protospacer, is then integrated into the CRISPR array in the bacterium's own genome, becoming a new spacer. When the bacterium encounters the same virus again, the CRISPR region containing the matching spacer is transcribed into RNA. This RNA molecule, known as CRISPR RNA or cRNA, then combines with another RNA molecule, tracrRNA, and the Cas9 protein. The cRNA guides the Cas9 enzyme to the target DNA sequence in the invading virus. If the cRNA finds a perfect match, the Cas9 protein activates its scissor function. Once the CRISPR-Cas9 system was understood and adapted for broader use, its potential in medicine became immediately apparent. Researchers quickly began to explore its applications in treating genetic diseases. These are conditions caused by specific errors or mutations in a person's DNA. Diseases like sickle cell anemia, cystic fibrosis, and Huntington's disease are prime examples. Early experiments in cells and animal models showed remarkable promise, demonstrating that CRISPR could indeed edit genes with a high degree of accuracy. Using CRISPR, scientists have successfully edited the faulty gene in blood stem cells taken from patients. These edited cells, when reintroduced into the body, can then produce healthy red blood cells. Clinical trials are underway, and early results have been incredibly encouraging, with some patients experiencing significant relief from their symptoms. Beyond single-gene disorders, CRISPR also holds potential for tackling more complex diseases, including certain types of cancer and viral infections like HIV. The speed at which CRISPR has moved from a basic science discovery to clinical application is astonishing. While the medical potential of CRISPR sparked global excitement, it also raised profound ethical questions, particularly concerning its use in human embryos. These concerns dramatically came to a head in November 2018. A Chinese scientist, He Jiankui, announced to the world that he had used CRISPR-Cas9 to edit the genes of human embryos. These embryos were then implanted, resulting in the birth of twin girls. He claimed his goal was to make the babies resistant to HIV infection by modifying a gene called CCR5. The father of the twins was HIV positive, and He Jiankui presented his work as an attempt to prevent transmission to the children. The announcement sent shockwaves through the international scientific and ethical communities. The primary concern was that He Jiankui had performed germline editing. This means the genetic changes he made would not only affect the twin girls, but could also be passed down to their future children and subsequent generations. The scientific community largely condemned He's actions as premature, unethical, and lacking in transparency. There were numerous criticisms of He Jiankui's experiment. The potential benefits to the babies did not seem to outweigh the significant risks associated with an untested and powerful new technology. Section 5. Healing versus enhancing, a blurry boundary. The advent of CRISPR technology has brought a critical ethical debate to the forefront: the distinction between using gene editing for medical treatment versus human enhancement. Most people would agree that using CRISPR to cure debilitating genetic diseases like Tay-Sachs or Duchenne muscular dystrophy is a noble pursuit. This therapeutic use aims to restore what is considered normal human health and functioning. However, the line between therapy and enhancement can become surprisingly blurry. Consider a condition like short stature. While some forms of dwarfism are clearly medical conditions with associated health problems, what about someone who is naturally shorter than average and wishes to be taller? What about enhancing intelligence, athletic ability, or even altering cosmetic traits like eye color? The concept of normal itself is a social construct that can shift over time and across cultures. Section 6. The perils of precision, unforeseen consequences. While CRISPR-Cas9 is often lauded for its precision, it is not infallible. One of the primary concerns is the risk of off-target edits. The guide RNA is designed to lead the Cas9 enzyme to a specific DNA sequence. However, sometimes the guide RNA might bind to other DNA sequences in the genome that are similar, though not identical, to the intended target. If this happens, Cas9 can make cuts at these unintended locations. Such off-target mutations could have serious consequences, potentially disrupting other important genes, leading to new health problems, or even increasing the risk of cancer. Ensuring the absolute specificity of CRISPR is a major ongoing research challenge. Another potential issue is on-target errors. Even when Cas9 cuts at the correct location, the cell's natural DNA repair mechanisms can sometimes introduce small errors like insertions or deletions of DNA base pairs when mending the break. Furthermore, there's the problem of mosaicism, especially relevant in embryo editing. This occurs when the gene edit is not successful in all cells of the developing embryo. The long-term consequences of any gene edit, particularly germline edits made in embryos, are largely unknown. Genes often have multiple functions, some of which may not yet be fully understood. Section 7. Beyond the genome, the expanding CRISPR toolkit. The original CRISPR-Cas9 system, revolutionary as it was, primarily functions by cutting both strands of the DNA. While this is useful for disabling genes or for inserting new genetic material when combined with DNA repair pathways, it's a somewhat blunt instrument. Scientists quickly recognized the need for more nuanced editing capabilities. This has led to the development of an expanding toolkit of CRISPR-based technologies that offer greater precision and versatility. One significant advancement is base editing. Unlike CRISPR-Cas9, which creates a double-strand break, base editors use a modified, nucleus-dead Cas9, dCas9, that can still bind to the target DNA sequence but doesn't cut it. This dCas9 is fused to an enzyme that can directly convert one DNA base letter into another, for example, changing a cytosine, C, to a thymine, T, or an adenine, A, to a guanine, G, without breaking the DNA backbone. This is like having a molecular pencil with an eraser, allowing for precise single-letter changes. Another powerful innovation is prime editing. Developed more recently, prime editing offers even greater versatility. Prime editing can make a wider range of edits, including all possible base-to-base conversions, as well as small insertions and deletions, with potentially fewer off-target effects and more predictable outcomes than conventional CRISPR-Cas9. Section 8. The future beckons. A world reshaped. Looking ahead, the trajectory of CRISPR technology points towards a future where our ability to interact with the code of life is profoundly enhanced. In medicine, the most immediate impact will likely be seen in the treatment of genetic diseases. We can anticipate a growing number of clinical trials and eventually approved therapies for conditions like sickle cell anemia, beta thalassemia, cystic fibrosis, and muscular dystrophies. Beyond monogenic diseases, CRISPR holds promise for tackling more complex conditions. Its role in cancer immunotherapy is already established and will likely expand, leading to more personalized and effective cancer treatments. There is also significant research into using CRISPR to combat infectious diseases, not just HIV, but also persistent viral infections like herpes or hepatitis B, and potentially even to develop new antimicrobial strategies against drug-resistant bacteria. Furthermore, CRISPR could revolutionize diagnostics, enabling rapid and accurate detection of pathogens or genetic markers for disease predisposition. The ability to quickly and cheaply read and write DNA will transform many aspects of healthcare. The prospect of designer babies, while ethically contentious, remains a persistent topic of discussion. As our understanding of the genetic basis of traits like intelligence, height, or athletic prowess improves, the technical capacity to attempt such enhancements could emerge. Section 9. Our evolutionary crossroads, choices and responsibilities. We stand at a unique juncture in human history, perhaps even in the history of life on Earth. For millennia, evolution has proceeded through the slow, undirected processes of natural selection and random mutation. With the advent of CRISPR and related gene editing technologies, humanity now possesses the ability to consciously and deliberately alter its own genetic makeup and that of other species. This is an unprecedented power akin to gaining control over the very script of life. It places us at an evolutionary crossroads, faced with choices that carry profound ethical weight and long-term implications for our species and the biosphere. The potential to eliminate devastating genetic diseases is a powerful lure, and one that resonates deeply with our compassionate instincts. The relief of suffering is a core tenet of medicine. Yet the same tools that can heal can also be used to alter traits for non-medical reasons, blurring the line into enhancement and raising the specter of a future where human evolution is guided by human desires rather than natural processes. If we begin to edit the human germline, making heritable changes, we are not just making decisions for individuals, but for all future generations. This is a responsibility of immense magnitude, demanding wisdom, foresight, and humility. One of the central ethical challenges is ensuring equitable access to these transformative technologies.